Hello students, today our topic of discussion is biotechnology, its applications and genetic engineering. Biotechnology is a modern form of industrial production that use living organisms, especially microorganisms. Some biotechnology processes involve naturally occurring organisms, but other involve organisms that have been produced by genetic engineering. Genetic engineering is a group of techniques which allow genes to be transferred between species. Genetic modification. This is possible because the genetic code is universal. Recombinant DNA technology is the set of laboratory techniques that combines genes from different sources into single DNA molecule. So you have now, I hope that you have understood to some extent biotechnology. Again, I am simplifying this. The technology making use of biological species. The technology making use of biological species called biotechnology, and for using these biological organisms there are some process. So this process have to be followed. So you can go through your textbook, you see page number 40, bacterial cell, where you will find the genes that are located either in the nuclear chromosomes, which are linear DNA sequence, and in plasmids, which are small extranuclear self-replicating circular DNA, as in bacteria, plasmids. So we will see in the next slide manipulating DNA. Now plasmid is one of the basic and very important part of DNA replication. So these figures are plasmids. This oval type figures, these are plasmids, and this is a bacterial cell. So plasmid is a small circular DNA molecule separate from the much larger bacterial chromosome. A plasmid may carry a number of genes and can make copies of itself. So plasmid has the capacity to copy itself. So let us see the next slide. The gene transfer. Plasmid, a small extra circle of DNA used to exchange genes. Now plasmid has the capacity to copy itself, also can be used to exchange genes. This is very, very important. The restriction enzymes, endonucleases, the enzymes that cut DNA molecules at specific base sequence. Some restriction enzymes can cut the two strands of DNA at different points, leaving single-stranded sections called sticky ends. DNA ligase, enzyme that joins DNA molecules together firmly by making sugar phosphate bonds between nucleotides. So gene transfer. So through plasmids, so gene is transferred from one place to another, one bacterial cell to another cell, recombinant plasmids. So plasmid is transferred to this cell, from this cell to this cell. So this gene transfer is done in this way, and that is also called genetic engineering. This is also one figure. This is sticky ends. You can go through later on properly, and later some examples of genetic modifications. We can find animals like goats, sheep, plants and many crops which can be genetically modified or improved. Then some rice, good quality rice, for example golden rice, and there are so many economic benefits of genetic modification. So we will not go again more. So I hope that this much is enough for today in this video. Thank you very much for listening.